Hello to all. My name is Gabriela Magossi. I am a PhD student from Kansas State University working with food safety. Welcome to this talk and thank you for listening. My presentation is going to cover my study on antimicrobial resistance of Salmonella enterica and Escherichia coli in the United States feed mills. A little bit of the reasoning for this research here. We have copper and zinc are metals commonly used in the swine feed industry. They're considered trace minerals due to being necessary for the animal's nutrition and are added to the feed during feed manufacturing. They are not toxic to mammalian cells, but have antimicrobial effects. Since these are heavy metals, these compounds can persist in the environment of the feed mill for long periods of time, even accumulating if not removed or cleaned. Also, sublethal doses of antimicrobials can lead to the development of resistance in bacteria. Previous studies have found co-occurrence of heavy metals and antibiotic resistance in microorganisms exposed to this type of environmental pressure or stress. Also, previous studies have shown the potential transmission of pathogens from farm to fork, or in this case, feed to fork. And we know food and meat products can become contaminated, and that contamination can be traced back to the animal during slaughter at the farm and even further in the feed. We all know how the rising antimicrobial resistance is a concern in human infections recently, since we have a limited amount of antibiotics available for treatment. Therefore, I was interested in looking into the environment of feed mills and using whole genome sequencing to identify antimicrobial resistance determinants in bacterial isolates. The objectives of the study were to identify antimicrobial resistance genes from feed mill isolates using whole genome sequencing, then determining the phenotypic susceptibilities of those isolates to antibiotics and metals, and finally to compare the antimicrobial resistance genotypes and phenotypes. The study design consisted of 4 states located in the Midwest of the United States. They were Kansas, Missouri, Oklahoma, and Iowa, all with large swine and agricultural production, where 6 feed mills were swabbed. The swab sponge samples were transported to the lab, where they were analyzed using selective enrichment in media for culturing. After culturing, the presumptive positives for both Salmonella and E. coli were then confirmed using specific PCR assays. The confirmed isolates yielded a total of 33 Salmonella and 30 E. coli isolates that were then shipped to the Center for Veterinary Medicine of the Food and Drug Administration, the FDA, in College Park, Maryland, for whole genome sequencing. For that, DNA libraries were prepared using the Nextera XT library prep kit and sequenced on either an Illumina MySeq or NextSeq sequencer seen here, depending on the availability. The genomes were then uploaded into the NCBI database, and the AMR Finder tool was used to match those genomes to referenced antimicrobial resistance genes, creating a genotypical resistance profile of all the isolates. Next, those isolates were subjected to phenotypical antimicrobial testing to determine the minimum inhibitory concentration of antibiotics using a sensititer NARMS gram-negative plate that contained this list of antimicrobials seen here on the right. They were amoxicillin with clavulanic acid, ampicillin, azithromycin, ceftriaxone, chloramphenicol, ciprofloxacin, gentamicin, meropenem, cefoxitin, nalicylic acid, sufixazole, and tetracycline. Heavy metals minimum inhibitory concentrations were determined using an agar dilution method. The concentrations used for both copper and zinc ranged from 0 to 40 micromolars. This was based from a protocol used by Amashavari and others. The results of the resistance genes, we found several genes encoding resistance to antibiotics. Salmonella had more diversity with 12 unique genes encoding resistance to 6 antibiotic classes. You can see here the aminoglycosides, others, which is phosphomycin, lincosamides, polypeptides, in this case, colistin, and tetracyclines, in addition to multi-drug resistance genes. Among E. coli, there were 11 unique resistance genes encoding resistance to 3 antibiotic classes: aminoglycosides, beta-lactams, tetracyclines, and also the NS-specific multi-drug genes. 33 isolates had PCO genes encoding copper resistance, and plasmids containing metal resistance cassette were present in all isolates. After a genotypic and phenotypic analysis, it was observed that Salmonella and E. coli isolates only express resistance to sulfonamides, tetracyclines, and ampicillin, while remain phenotypically susceptible to the other antibiotics tested. A total of 7 isolates had the gene SUL1, which encodes resistance to the class of antibiotics sulfonamides. And we observed that 9 isolates expressed phenotypic resistance to sulfoxazole out of the 63 isolates, with a significant correlation between genotypic and phenotypic resistance for sulfonamides. A total of 23 isolates had at least one of the tetracycline-resistant genes, TET A, B, or C, while 25 isolates expressed phenotypical resistance to the tetracycline antibiotic. Again, there was a significant correlation between genotypic and phenotypic resistance. And interesting to observe, in both cases, there were 2 isolates that expressed resistance to the antibiotic but had no matching gene, which might indicate that there is another genetic element to confer resistance to those drugs. In addition, there were 3 E. coli isolates expressing phenotypical resistance to ampicillin without displaying any resistance genes for this class of antibiotic. There were also phenotypically resistant to tetracyclines and harbored these genes shown here. This image is an example of E. coli plasmid showing the BLA genes are close to the APH genes, all present in those isolates. Maybe the unknown gene for ampicillin can also be found within this plasmid. It is a hypothesis for future research. All isolates that had the PCO genes that encoded for copper resistance were tested for phenotypical copper and zinc resistance. The mean inhibitory concentration was found to be 20 micromolars for copper, seen here on the top picture, and 5.2 micromolars for zinc, observed here on the bottom pictures. No zinc-resistant genes were identified in any of the 63 Salmonella and E. coli isolates tested in the study. These observations are similar to other studies evaluating heavy metal resistance, where minimum inhibitory concentration for copper resistance was around 20 micromolars for the bacteria that is expected to express resistance, and around 6 for those that were not expected to express resistance. In summary, the conclusions for this study are that we are able to show the presence of genotypically and phenotypically antimicrobial resistant bacteria within the swine feed mills tested. And interestingly, there were bacterial isolates that were capable of expressing phenotypical antibiotic resistance without having known resistance genes for that specific class tested, which leads to the speculation that there is another gene or combination of genes associated with that expression. Antibiotic resistance was not expressed phenotypically for the majority of antibiotics tested, the phenotypic dormant isolates. That can be due to the loss of resistance encoding plasmid, or possibly they would only express resistance while under stress, which was not the case during this study. And finally, we could determine that feed mills can potentially play a role as an entry route of antimicrobial resistance into the animal production industry and the human food chain. And that concludes what I wanted to share with you. Thank you so much for listening.